Okay, so we'll continue with the next chapter now. And in this chapter, we're going to look at the real data set, and we'll try to quality control the cells and the genes in this data set and remove all the cell outliers and gene outliers from this data set. The data set is the same as you used in the UMI chapter with TUNC data set. So let's start with the analysis. And again, we'll be using the scatter package that Davis introduced in the previous chapter because we find it very useful and practical for single cell data. So we first load the data, which you probably have already done. And then we can have a look at the molecules file and the annotation file as well. So if you look at the molecule files, so there was a question, what we have in columns, column names and row names in this matrix. So the column names are the cell IDs. And this is one of the requirements of the scatter package, that your cells have to have unique cell IDs. So they have to be unique. And this is very important because then you can actually get information about every specific cell without confusing the cells. And then row names contain ensemble gene IDs. And the values inside the matrix are the number of UMIs in each cell, in each gene. If you look at the annotation table, which is the next one, this is basically the metadata that you have with your data set. And here you have association with of your cell ID. So you have the same unique cell IDs in this column here. And then it contains a lot of information about each cell. So basically, this table serves you as a annotation metadata for your data set. So you can actually have a lot of more information about each cell in your data set. You can actually create the same table for your genes. For example, you can store the gene length information or something like that. And then you can actually import all these annotation tables together with the data table in your scatter object. So that's what we're going to do now. So here we create the phenodata variable, which contains information about the cell. We create it from the annotation table. So we need to do this because, as Davis mentioned, we work in the bioconductor universe. So we need to basically satisfy some classes of the bioconductor here. So we create the annotation table. And so this phenodata contains, as I said, information about each cell. And then you can actually create this new scatter object using new SCSset function. And we say that the count data is the molecules, UMI molecules that we have. And the phenodata, which is the cell information data, is the phenodata data frame that we had before. And this should work without errors. And then basically you have your scatter object ready. And then you can investigate and do quality control of your cells and genes. So first of all, we will remove the genes that are not expressed in any cell because they are kind of useless. Those genes have zeros across all the cells, so they won't give you any value in the analysis, in any analysis that you do. Then we also define the control features. So the control features can be either ERCCs that were introduced by Martin in the previous chapters. So we know that in this data set that we have, ERCCs have names which start with ERCC. So we basically just say all ERCCs from this data set contain this tag in the beginning. And then we also know that which genes are mitochondrial genes in this data set. So we define mitochondrial genes manually. This is taken from the publication, so we just took the author's advice about the mitochondrial genes. And then in your scatter object, so then basically we run the calculate QC metric. So once you created your scatter object, you can run this function on your object, and it will calculate lots of different values for you, for each cell and for each gene. But when you run it, you can also define the feature control. So you say, I want to calculate QC metrics, but please also use this variable as ERCCs, and this variable as mitochondrial genes. So basically then Scatter will use separate calculations for your ERCCs, for your mitochondrial genes, and for endogenous genes as well. It will be separated, and then you can compare what you have in your RCCs to what you have in endogenous genes. So now everything is ready. So we have created the scatter object. We provided information about the cells. We told the object what genes are ERCCs and what genes are mitochondrial genes. And we run the calculate QC metrics function. So we're ready to go to explore the data set, the object now. So first of all, we're going to look at the library size. So the first step, which is probably the most reasonable, is to look how many total counts you have in each cell. So what we do here, we plot a distribution of the total counts in all the cells. So this information is contained in the total counts column of your scatter object. And as you can see, we have this distribution here, which looks like normal, but there is kind of tail on the right. And so basically here we have a couple of exercises. So the red line here is, we define it manually, which is 25,000. So what we want to show by this red line is that the cells that are on the left of this line contain less than 25,000 total counts, right? And because majority of the cells in this data set, as we can see, contain about 80,000 total counts, we probably want to remove them because the number of counts in those cells are quite low. We don't know why. It can be any technical reason, but we probably don't want to consider those cells in the downstream analysis. So this is defined manually by us. So you can actually find your own threshold. And for your own data set, you probably will have a different distribution. So we just want to show that basically once you start doing quality control analysis, there is no automated procedure. So you have to look at everything yourself, and you have to decide which cells you remove, which cells you keep. So let's look at the exercises. So exercise 1 is how many cells does our filter remove? And the second question is, what distribution do you expect that the total number of molecules for each cell should follow? So the first question you can work out yourself, and then you can look at the RStudio file if you want to. For the second question, so what do you think should be the ideal distribution? If we had an ideal data set, for example, perfect data set, what should this distribution look like? Any suggestions? Simple ones? Normal distribution. Yeah, so we think it should be normal if the data set was perfect. But as I said, there can be technical problems in sequencing, technical problems in library preparation. And that is why some of the cells are basically in this tail of your distribution. And actually, we have lots of cells which are in this tail, which are probably also should be removed, in my opinion, but we decided to keep them. So it's your choice, and you have to, I would say, if it looks like that, cut the tails and keep the majority of the cells. But you have to look at each cell separate. Like here, we know that the total counts is very high. Probably you should go and look in the cell and see what's wrong with it, and why is it so high. So the answer to the question 1 is this. So if we set the cutoff at 25,000, we can remove 46 cells from our analysis, but we still keep 800 cells. So this is actually normal numbers here. So usually if you have hundreds of cells, if you remove a dozen, a couple of dozens, it's fine. If you have only 50 cells and you remove 46, this is not fine. So you have to figure out what to do with your own data set. So let's move on to the detected genes now. So we do the same procedure, but now instead of total counts, we use total features column from the scatter object. And we plot the same distribution, but of the total features. So the total features, what it means is that it's the number of detected genes in all cells, which is the number of genes, the expression of which is higher than 0 in your data set. So we can see that in majority of the cells, we have about 9,000 genes expressed, right? So basically, it's only, as you can see, it's only half. If you have in total 20,000 genes, so in majority of the cells, we only have 9,000 of them expressed. Everything else is 0. And again, we have this heavy tail on the left containing the cells in which the number of detected genes is much lower than the majority of the cells. So most probably, some of these cells actually are the same as the cells that we removed before. We haven't actually removed those cells yet. We created a filter, which we'll use in the future to remove the cells, but we haven't removed them yet. So this is, again, the total data set. And that's why some of the cells may be actually the same. So if you remove them before this step, probably your tail will be different. But we just want to show that if you start with this, that's what you see. And again, probably for the ideal data set, you would want to have a normal distribution here again. So our advice is to cut the tails. But here we don't have actually, here this is a nice tail, so I wouldn't remove it. But this one is quite bad. So with this cutoff, we can remove 120 cells. But again, we keep 750 cells in our data set. So to me, this is also fine. So 10 to 20% of the cells, if you remove, it should be fine. Don't be afraid of doing that, because then your downstream analysis would be much more robust, and you will get more biological signal instead of some noise. All right, so we have looked at the total counts, total features in every cell. Now we'd like to look at the controls. So we have ERCCs and mitochondrial genes as controls. We want to have a look how the counts in those genes look like. Right? So first, we plot the percentage of counts in mitochondrial genes. So again, as we said before, scatter is very nice. Yes, question.
If you have a heterogeneous data set of different cell types, wouldn't you have to take that into account when you just cut off? You might be cutting off the whole cell type because there's
Yes, so the question is if your experiment is very heterogeneous, so you have different cell types, lots of biological signal, and this can affect your distribution, the one that we saw before. Right, so basically, those cells can be actually meaningful. As I said, each experiment is different. So if you know that you have cells which might contain less counts because of the biological reason, then you should keep them. If you think that your cells should be all the same, you should remove it.
Is there a way to do this with a multimodal distribution? Is there a rule for it, or is it just?
There is no rule for that. It's just manual picking of the cells. At the moment, I think the experimental techniques are not perfect in a way, so you have to do it yourself manually. Maybe in a couple of years there will be some method which will be quite robust and stable, but for the moment there is no automated procedure for that. Sorry? So the question is, yeah.
You still have to eyeball it. So each experiment is different. There's no
Yeah.
there's no way to automate it.
Yeah. So basically you just, it's not a question, right?
Yeah.
All right. Yeah, okay. Yeah, so but I mean, if you have a bimodal distribution, so if you have like this peak and then a peak here, definitely there is something interesting or something really bad. So you have to look at it again. Yep.
Yeah, there's a function instead that lets you, so for a given metric, detect outliers based on deviations from the median.
I think do we cover it?
I can't remember.
We have some, actually we have some automated procedure for cell outliers, which I'll show you later. But you have to be careful because this is just, you know, it's not, for each data set it can't predict the outcome, I think. Okay, let's look at the mitochondrial genes. So with the scatter, it's really easy to plot your controls. So you just say plot FINA data, and then you define a parameter here. So you want to plot total features, which we saw before, against percentage of counts in feature controls mitochondrial genes. That's what we defined when we created the scatter object, that controls are either ERCCs or mitochondrial genes. So here we want to look at total features against percentage of counts in the mitochondrial genes. And basically with one line of code, you can easily plot these things. And you can also color, or you can change the shape of your points by providing extra parameter here. So here each point is a cell, because we are plotting FINA data, and FINA data is the information about the cells. So each point is a cell. On the x-axis, you have total features, which is the number of expressed genes, as we saw before. The average is about 9,000. So it's about here. So you can actually see the same distribution from top of it. For example, we have majority of cells here, and then we have this tail that we saw before. And then the y-axis shows you the percentage of counts in feature control, in mitochondrial genes. So we see that majority of the cells have like 5% of the counts in mitochondrial genes, which is fine. But some of the cells, like these ones, they have about 15% of the counts in mitochondrial genes, which is probably not very good. If you go over this, if you like have 20 or 50% of your counts in mitochondrial genes, that means that something went wrong with your cells. Something like the cell starting dying or doing something wrong. So that's the purpose of the controls. Basically, you want to see how many counts you have in your mitochondrial genes. And usually when you have a lot, it means that something went wrong with your cell. So here I think we keep, so we keep this, maybe we don't keep, I don't remember. So we'll see afterwards. So you can either set a threshold here, so additional cell filter, and you say, if you have more than 15%, we cut it off. Or you can say 10%. You can also cut off before that. So, yeah.
Sorry, is the term that
Yeah,
We didn't define that earlier, did we?
we didn't define. So
Is that
when we
in the scatter package?
so when we run the calculate QC matrix function,
Yeah.
and we said we want to use these genes as RCCs and these genes as mitochondrial genes, this function actually calculates lots of things that you then that shows you on the plots afterwards. Yes. So when you calculate QC, it takes some time, but then everything is calculated. So when you plot it, it's ready to go. So it doesn't calculate anything when it plots it, I think. And you can actually look at these names. So if you say phenodata of your object, and they say call names, it will show you all the call names that you have there. And you can explore manually everything there. So then we do the same for the ERCCs. So basically, instead of mitochondrial, now we say we want to look at ERCCs. And now we can actually see that there is something wrong with some of the cells. So majority of the cells, as you can see, don't go over than 6–7%. So the number of counts, the number of UMI molecules in these cells is less. In ERCCs is less than 7%. But there is one batch, orange batch, this one, where you have lots of counts in the ERCCs genes. And this is another sign of the bad cells. So if you have lots of reads in your ERCCs, that means again that something went wrong. So it's better to remove those cells. So that's the exercise 3. We want to create filters to remove the bad batch. It has this name. And we also actually, yes, we remove the cells where the counts in mitochondrial genes are more than 10% of the total counts. So we create these 2 filters based on our controls. And then, yeah, you can work this out or use the RStudio file again. So these genes, these filters remove 96 and 37 cells correspondingly. So let's look at the exercise 4. The question is, what would you expect to see in the RCCs versus counts plot if you were examining a data set containing cells of different sizes? For example, normal versus senescent cells. That's related to the question of bimodality of the distribution. So if you know that, like, how ERCCs work is that you add the same amount of ERCCs in every cell, right? So what happens is that if your cells are of different sizes, so you have one population of cells which are small cells and the other population is bigger cells, but you add the same amount of ERCCs to every cell, the concentration of ERCCs in larger cells will be smaller, and that's why the number of reads in those cells will be smaller. So you would expect that you would have 2 different distributions of ERCCs on the plot, and you should not neglect the one of them, because you know in advance that you have 2 populations of cells with different sizes. So that, again, coming back to the same question, because you do the experiment, you know the biological details, you have to take them into consideration when you quality control your data set. Okay, have you finished with the exercises? Shall we move on? Okay, so now we have created 4 filters and we want to apply those filters on our data set. So we do it this way, so you just create additional column in your scatter object, and then you just say you apply all the filters, because those are logical vectors. You basically just AND all of them together using AND expression. And this new column use will contain basically all filters together. And then if we look at this combined filter, we actually remove 200 cells from our data set. But we still have 654 cells. So 200 is a large number, but as you saw before, we have one batch, which is completely different from everything else. So it's worth removing it. I mean, this is a large number. Usually you would want to have something less than that. But you never know what can happen in the data set, so it's better to remove everything which is suspicious. So Scatter also contains default filter. So if you don't want to bother with looking at the figures or plotting the histograms, you can just use the Scatter default features. So again, in the same, that's related to the question about the names of the different parameters. So in the same table where you have information about the cell, after the quality control step, Scatter will create these columns with these names. And you can see the description of each column here commented. So basically, it filters on total counts for you automatically. Basically, this filter removes cells with unusual number of genes. So maybe Davis can comment on that. I don't know the details. You can actually go to Scatter documentation and read what these values are. So if you don't want to bother with doing manual quality control analysis, you can do this very quickly.
Yeah, I guess the point I'd make about those filters is the defaults are very lenient.
Mm-hmm.
So yeah, like you can see, like less than 80% of ERC spiking is the sort of default filter on that. So that's extremely lenient. So
yeah, so these are very weak thresholds, as you can see. So this is just to remove something really bad, right? Yes.
yeah, so you can tweak the options in there if you want to say, I want 20% or whatever else is there.
Okay.
There are arguments that we haven't listed in there, but if you check the documentation, you can find those arguments and change them.
Yeah, so you can control these thresholds when you run calculate QC function. But as you can see, this filter removes only 6 cells. So it doesn't really find the bad batch because it's quite weak, as we said, the default values. Also, there is an automatic filter, automatic procedure to find outliers in Scatter, another one, which is basically, instead of looking at the genes, it uses the calculated values, the calculated QC values, and it plots the PCA plot based on only these values, and then tries to find the outliers on the PCA plot. So if we run this, this is the code to run it. So you define So I think, yeah, if you provide this, you say PCA data input, and you say P data. So then it will plot PCA based on the values in your PhenoData object. And basically, once you run it, it plots the PCA plot. And it can also automatically detect outliers on this PCA plot. So with the orange, it shows you the outliers, and the blue cells are fine, according to this filter. But again, this is all automatically done. So like here, it actually defined outliers quite nicely. And probably those correspond to this bad batch that we saw before. Some of them are actually still probably outliers. But you have to look at this plot and decide whether it looks okay or not before using it. So this filter removed 73 cells. So the last exercise related to the cells is, let's compare all the filters that we created so far. So we have an answer here already, but no code. So the code is in the RMD files in RStudio. So you can use, we used Venn diagram function from Lima package to create this plot. So if you want to do it, you can do it yourself, or look at the RMD file. And we wanted to see whether these filters overlap with each other. So which cells do they remove in common and which cells are not. So as we can see here, our manual filter, the one that we created in the beginning, actually contains all cells that were removed by the automatic filters, right? So our manual filter was actually better, well, stricter than the automatic filters, but also it didn't miss any cell that the automatic filter detected. Which means that probably it's better to do it manually and be sure that you have selected only the best cells for your further downstream analysis. Any questions?
Just to clarify, the filters are just which commands with some UMI code runs below some.
Sorry?
The filters are created by just which calls for, say, UMI total counts and then plus them whatever your threshold is?
Essentially, yes. So these are all hard thresholds.
Okay.
Yeah. You could obviously be more subtle.
I'm just worried about the format of the filters. They're literally true, false.
Yeah, so anything that produces the appropriate true, false.
True, false set.
Well,
Although
so
they could also be
0, 1,
well, so
so
if you add it as a column in the SE set object, then it needs to be a vector of the same length as the number of cells. So in that case, indices that are true, false work perfectly. To subset any other way, you can also just provide indices, you can provide true, false indices, you can provide cell names to subset anyway. All of those will work. So.
And the cell names would be put into this UMI used part.
No, so
So-
that wouldn't work.
So I think the question is how we define the filters.
Yeah.
So I think we define them using logical vectors, but actually you can define them as you want.
Yeah.
It can be
Well,
0, 1s, right?
If you want to add a column to the phenodata as used, then that needs to be either true, false, or 0, 1. Like it needs to be a logical vector of the same length as the number of cells, or else that attempt to assign that will fail because it's not at the correct size.
Yeah, but for us it's why we use the logical, because when we merge them together, all the filters, you just say and basically you don't need to do any more operations other than that. So that's
But of
easier.
course, in such a command, you could use or statements or any combination.
Yeah.
Generally speaking, if you're subsetting an SCE set object, then you can use, say, on the cells, and you can use logical indices, you can use names of the cells, so cell names, sample names, or you can use integer indices, and all of those will work correctly to subset.
Yeah, that's the same as the matrix or data frame, right? The same, more or less the same thing. Yeah, so you can manually subset your cells from the scatter object, or you can create a filter, either logical or not logical, but then when you use the filter, you have to say either you look for true or false values, or you look for 0, 1s values, whatever you defined before. Okay, so let's move to the genes quality control. So we have removed the bad cells, created the filters, that's fine. Now we should look at the genes as well. Maybe there are some genes which are bad in our data set. So Scatter provides these functions, function plot QC. And if you also say type equals highest expression, it will show you the genes, the top 50 genes, and they account for 15% of total counts. So basically, in those 50 genes, you have 15% of total counts of your matrix. And you can see they're ranked. So you start with the most highly expressed gene. Basically, for example, here we have ensemble gene IDs, which are not meaningful, but if you have gene names instead, you would see which genes are the top highly expressed genes. And we can see that we have ERCCs here at the top, which is also reasonable because usually you have quite a lot of genes in your ERCCs. So this is just an overview of your highly expressed genes. But then we want to create gene filters, right? So first of all, we would like to remove genes whose expression level is considered undetectable. So we define a gene as detectable if at least 2 cells contain more than 1 transcript from the gene, right? So if you look at the gene row in your matrix, you need to have at least 2 1s in this row. Then we say the gene was detected. If it's only 1 1 or 1 something else, it will be removed. So you can set this threshold yourself. Again, it's better to be stricter than weaker. So it's your choice, basically. So here we say if we were considering read counts rather than UMI, a reasonable threshold would be at least 5 reads in at least 2 cells, because usually you have the number of reads is usually much higher than the number of UMI counts. So it depends on your values, on your units. So be careful with this threshold. But again, if it's strict enough, that's fine. So here we just create this filter manually, the one that I described to you before. And if we look at this filter, we can see that we removed about 4,500 genes, but we still keep 14,000. This is fine. So I think if you go down to 10,000 genes, it's also okay. So usually you can remove quite a lot of genes from your data set before doing downstream analysis, because most of them will contain lots of zeros. And I think that's it about the gene filter. And the last thing we need to do is to save our data. So now we have this use column and use row, which contain combined filters for the cells and combined filter for the genes. So if you look at the data set, when we apply those 2 filters, we will see that we removed 4,500 genes, and we removed 200 cells. So this is our final data set after the quality control. So we have one extra step here. We create a slot in our object. So this setExpress function, it can create a new slot in your object, which will have the same size as your count matrix. So the same number of cells, the same number of genes, but it will have a different name. So we name it log2 counts. And we just basically write a log transformed count matrix into this slot. So we do that for the course purposes, basically, because as Davis mentioned before, it's better to do log transformation on your data before visualizing it. You want to make it a bit more normal. And because of the data properties, usually you might have some genes which are very, very highly expressed. So if you don't log transform, your PCA plots will not look ideal. So usually that's the minimum thing you need to do with the single cell RNA-seq data, is to log transform the data. But that's not the most perfect data to look at. So after, how to say that? So log transform is the minimal thing you need to do, but you also want to do some additional things like normalization by library size and stuff, which we'll show you later. But this is the minimum thing, and we want to do it now before saving the object. And then we just save the object to this folder. You already should have it in your RStudio files, so you don't need to save it, or you can save it again if you applied all the filters before. And basically now we are going to do the next exercises, which is the next chapter. So we're going to do the same thing, but now using the reads instead of the UMI counts. So you will be able to compare the reads, the read quality to UMI quality. And you will be able to see how they look, if they look different or not, and what are the differences between them. Yes?
Most of the QC and things you're showing are based on UMI based protocol. If you have full length protocol, as you said at the start,
Yes.
what would change? You won't have the UMI counts at all, right?
Yeah, you would have reads, and that's what you're going to do now.
Or you just have the reads, right?
Yes. So now we take the reads file. So for this data set, the cool thing about this data set is that it has both UMI counts and read counts. So they did both things at the same time or differently, I don't know. So anyway, they have both of them. And it's also quite balanced. So the batches that we saw before, they were basically mixed properly so that you can be sure that if one batch is an outlier, then it's a real problem. It's not that the batch was done on a different plate or something. So it's a very balanced experiment, and it contains both UMI and reads. That's why we're using it in the course. So the next chapter, expression QC reads. So we think we should do it yourself. You can go through it, make on all the plots. Again, you have the RMD files. You can just run the RMD files if you want. But the purpose is to compare the UMI QC to the read QC and understand the differences between them. So we'll come back to the exercise after 15 minutes. So you can work on your own, and then we'll look together at the results.